Welcome to the laboratory of Dr. Heike Daldrup-Link.
We would like to thank the California Institute of Regenerative Medicine for funding this project.
Labeling stem cells with fluorescent contrast agent allows for an in vivo tracking of transplanted stem cells in vivo. Subsequent optical imaging of the labeled stem cells permits longitudinal studies where one is capable of non-invasively monitoring the migration of the cells in vivo. This video will show you how to label both human embryonic stem cells and human mesenchymal stem cells with fluorescent dyes.
Hi, I'm Sophie Boddington from the Contrast Agent Research Group in the Department of Radiology at the University of California at San Francisco. Today, I would like to show you two different techniques for labeling stem cells with fluorescent contrast agents. One for labeling mesenchymal stem cells with the fluorescent dye DID, and another for labeling human embryonic stem cells with indocyanine green. So let me start by showing you our technique for labeling human mesenchymal stem cells with DID. To begin the procedure for labeling mesenchymal stem cells, we have to trypsinize and count them to get a suspension with a defined number of cells. Since this is standard tissue culture technique, we will not show it in detail. We want to suspend the cells that we will label at a density of 1 million.